the title, Interactions Between Microscale Physical Processes and Soil Biochemistry and Biology. This is like a general big topic of one of the key topics that my lab works on. And what is the big idea behind the interactions, this focus on those interactions, is that all soil chemical and biological processes, they take place within a certain physical framework. That's what soil solids and porous, that's what they create, where the chemical fluxes, biological movements, where all those things take place. And performance of chemical reactions or biological activities, it is regulated by what can flow through those pores and where the access to the resources and the soil solids can be accessed. Studying this interaction between physics and biology in terms of the mechanism, the fluxes, the transport, the movements, is especially important if we consider the challenges we're facing as far as future climate, because it's only if we understand the processes that we can properly model those processes, meaning if we understand and model, then we can hopefully predict better what will happen as the current conditions change and climate change. Some of those specific topics that we worked on in the past few years, decomposition of plant residues in the soil. We're focusing on again, those specific process-based mechanisms through which soil physical characteristics, soil pores, influence the rates of decomposition, what happens with the carbon that comes from those plant residues, how the emissions of CO2 and nitrous oxide coming from those decomposing residues will be influenced. We now moving into studying root growth, how growth of the roots, the architecture of the roots, the carbon that comes from the root, how those things might be influenced by the pore characteristics that the roots encounter when they grow through the soil. Again, what that means to the carbon that is being added by the roots to the soil. The losses of that carbon is CO2 and nitrous oxide. The big and very interesting topic is, of course, microorganisms, because for them, those soil physical structure creates the habitats where they actually live and influences the microenvironmental conditions within the soil that will directly influence what will happen to activity of those microorganisms, what resources they have, how much oxygen or no oxygen they're getting, can they communicate to each other, can they be affected by the predators. All those things are influenced by the physical structure that enables movement of things in the soil. And of course, in that part of our work, we heavily rely on microbial ecology colleagues and microbiology colleagues, as that's way beyond my expertise. All those fancy microbial stuff, very fascinating ones. And so the big ultimate goal is what all this means for soil carbon protection and soil carbon gain. As probably many of you are aware that in the last 10 years, there has been a paradigm shift in what we now believe is most important for soil carbon gain. So that while for many decades, people thought that it is uh, the what limits what's important for increasing soil carbon is to increase the quantities of recalcitrant materials that are added to the soil. But now we're starting to very clearly understand that there is really nothing that microbes cannot decompose. If there is the right microbe with the right tool, they can eat anything, including plastics. It's just the tools I needed there. So what really is now considered to be a big driver in how much carbon is being protected is the accessibility. And of course, physical structure, physical framework, pores, is what makes carbon accessible or not accessible to microbes and their enzymes. So what all this our focus on physical framework makes life very difficult for us as far as the experimental work and what we can and cannot do. Because clearly, if we want to study physical framework, we cannot destroy it by sieving soil, because then what do we study? And it's not that we just came up with the importance of idea of studying soil intact. That has been known and recognized for a really long time. It's from 1938, a statement from a very well-known soil scientist that crushed or pulverized soil is related to the soil formed by nature, like a pile of debris to a demolished building. And that's, since then, this theme of importance of soil being studied intact just been resurfacing again and again. One does not infer the operational efficiency of a hospital by studying bricks into which we demolish it. And typically, the undisturbed profile in reality exists as a continuous convoluted core bounded by solids. So what you see here on the left is an illustration from a recent paper by colleagues of mine, where you're looking at soil profile, which is basically that intact soil body. You can separate it into aggregates, and from topsoil, that's will split into those mass of small particles, aggregates, variety of ranges. In the subsoil, in this particular profile, the argument is that the stronger force you apply, the smaller aggregates you will get. So if you just apply a light force, you get those big aggregates. You start increasing it, you get smaller and smaller aggregates. So what do you study at the end when you study the aggregates? And on the right, what you see is the image of soil sample from T7 from the main site here. And, maybe you can see aggregates there, I cannot. And if we just keep looking at the image and the force inside it, it indeed just one big blue convoluted pore space that connects everything within that particular small soil sample, but can be easily seen how that would extend to the entire soil profile. So what this focus on studying soils intact, what that means for us, it means that there is a lot of very hard work into taking the intact soil samples, and Maxwell is particularly affected by by that, as he's our expert on intact soil sampling and done a lot of poking a lot of holes in KBS LTER soil so far. So then what after that, what we have to do is to deal with all kinds of national labs where we can take our intact soil samples for scanning so that we could see using, well, computer tomography is one of the tools. We can actually see inside and see things as what I was showing on that previous image. And that is not, well, I mean, it is fun eventually, but jumping through all of those loops of getting into those labs and trainings and safety, it's very luckily now there is a scanning facility at MSU as well, and so we closely collaborate with Michelle, who is very helpful in getting some of our needs in looking at soil intact addressed. But the most challenging part begins after the samples are scanned, because in order to convert the images into useful, something that would be useful information, it takes a lot of time and effort. Can take hours, days, weeks, as Ricardo is now experiencing, to actually generate, segment those pores, segment pieces of organic matter in the soil. So it's, yeah, that's challenging life that we have. One other, but just one big benefit of looking at the soil in intact fashion, and also using those fancy and time-consuming and difficult to use, but still very imaging tools, what it pays off, especially that I want to emphasize, is analysis of what happens with plant residues and their decomposition in the soil. Because what we can do, we can actually see how the piece of root or piece of plant residue, how it looked originally in that intact body of soil, and what happened with that after the incubation. We can find the answers without actually destroying the soil and pulling that residue out. One point I want to make is that is especially important for roots, because when root grows through the soil matrix, it modifies its environment. It changes bulk density around it. It changes rhizosphere soil properties. It changes microbial communities. It changes how much carbon that soil in its proximity will have. And then when the root dies and starts to decompose, it decomposes in that special environment that it's created. And so what we often have to do is take roots out and then mix them with the soil to study root decomposition. That might not give us a very accurate picture of what actually happens with roots when they decompose in situ. So with the CT scanning, we actually able to see what happens with roots in situ, but of course that requires a lot of forethought as far as planning the experiment. So one and a couple of last things I want to mention is that we try to use this fancy CT scanning information to pique people's interest in soils, especially general public. So there is example of one of our activities when we participated in exhibit at Michigan Science Center 2 years ago. That purplish top picture is actually from our portion of the exhibit. So with, I don't know, unbelievable quantity of time into preparing that exhibit, but it was really nice to see public interest in soils and roots and plants. And this is an ongoing thing at MSU museum right now. It's 1.5 Celsius. That's the name of the exhibit. And so Maxwell and Jinho and Ovia, and who works in our lab, they did an unbelievable job in creating a piece for that exhibit, and so that just really encourage you if you're on campus to check it out. Well, not just their piece, but also that whole exhibit has a lot of very fascinating components there as related to climate change. Okay, this is all I had to say, and now let me turn to the people who actually do the work.
Hi, my name is Jino Lee. I'm a PhD student in the Kravchenko lab. And the topic of my part is the annual development of beneficial pore structure, as I described, and the carbon characteristics of the soil. As you already know, that the prairie vegetation is known to have the benefit for soil carbon gains because it has extensive root systems with a greater below-ground biomass compared to that of regular annual crops, and even compared to that of other perennial vegetations. So it released large amounts of active carbon into the rhizosphere. So it leads to greater amounts of the carbon input into the soil. And that large amounts of the carbon will be rapidly processed due to the fast microbial growth and turnover promoted by the prairie vegetation. So I'd like to say the prairie vegetation is the, potentially beneficial for the long-term carbon stabilization. And soil pores also have a role in the soil carbon processing and soil carbon gains. The left rotating the images are the arrangement of the solid and the pore, solid phase and the soil pores, which was segmented by using the X-ray micro CT scanning, as Sasha described. And especially the pores within 50 to 150 micron size provide a space for the microorganisms to live and thrive. So that microorganisms are responsible for the decomposing organic substrates and converting it into the soil organic matter. And we recently found from MLE sites that the prairie vegetation have had a higher volume of, a greater volume of that pores compared to that of the switchgrass. And also we found that the amounts of the amounts of the microbial biomass carbon and the content of the soil organic carbon was also higher in the prairie vegetations than those in the switchgrass vegetations. So we still have the questions. How quickly that prairie vegetation develops the beneficial pore structure and starts soil carbon gains. So we hypothesize that the prairie vegetations will gradually increase the volume of such pores, microbial biomass carbon, and total organic carbon content of soils. And it will eventually have the greater volume and the greater content of the soil than that in the first year. And since we collected the samples from the prairie strip, we also have one more questions about for the adjacent agricultural soils. Will that positive influence from the prairie strip extend into the adjacent tilled agricultural soils? We are thinking that the prairie vegetation, prairie strip will bring those benefits into the adjacent crop field. Because the microorganisms so that beneficial microorganisms will spill over via the runoff after rain and even through the hyper stretch of the roots from the prairie vegetations, and also that prairie strip will enhance the plant available nutrients. So it increased the overall soil health and productivity of the field, and it lead to increases in the crop yield and also carbon sequestrations. And other expectation is that the prairie strip reduce the soil erosions, so it can prevent the carbon-rich topsoils from being washed away. So we hypothesize that that agricultural soils adjacent to the prairie strip will have greater volume of such pores and greater content of the carbon than far away from the prairie strip. So for the questions, we collected samples every year since 2020 April. The samples from the first year were collected right after the wheat cropping, and the next year we collected sample after corn cropping, and again we collected sample after the soybean. The first year the samples were collected 1 year after the prairie strip installations and the switchgrass the planting in the T6 field, T6 plant. And we chose two sampling locations in the T3 plots. One from the center of the prairie strip and another one from the location 2 meters apart from the edge of the prairie strip. And we also collected one set of the sample from the T6, the center of the T6 plot. We collected two soil cores and one surrounding loose soils, and we scanned the cores to get the pore size. Scan and analyze the cores to get the pore size distributions, and particularly to measure the volume of the 50 to 150 size pores. And using the loose soils, surrounding loose soils, we also measure the microbial biomass carbon and total organic carbon of the soil. So here's a research of the pore size distributions and the pore volumes of that size range. But since we have processed just 6 of 12 replicates in the first 2 years and only 2 replicates of the third year, this is not the incomplete data, but the left figure is one example of the pore size distribution. The y-axis indicated that the pore fraction in the entire images, entire volume, and the x-axis is corresponding the size of the pore. Since we are interested in the pore volumes within the 50 to 150 micron size, we merged the pore volumes within that size range and calculate the pore volumes. We found that in the prairie strip and the switchgrass vegetations, that pore volume increased for the first 2 years. Then it tends to maintain, even though it's only from the 2 reps. But the volume of such pores in the crop rotation field did not increase for the observed 3 years. And we also found that the microbial biomass carbon increased in the prairie strip and the switchgrass vegetation for the last 3 years, while the microbial biomass in the crop rotation field is relatively steadily increased for the 3 years. And total organic carbon content slightly increased in the prairie strip and in the switchgrass vegetations, while the carbon content decreased in the crop rotations 2 meters away from the prairie strip after the year of the soybean. We also try to compare those data with the data currently we have. For example, the carbon contents data, which was measured in 2013, and also we have the data from the Lux Arbor, which is not that far from this KBS field. We found that the carbon contents of the prairie is almost similar to the levels of the Lux Arbor site contents, but we couldn't find any similar contents to the other fields, such as the KBS field and KBS T3 field and the KBS T6 plots. Since it's still ongoing, since it's the ongoing research, and even though it's collected from the same field, that should be that have the different histories of the site, and also forgot the last reason. It was important as far as I remember. Yeah, they should have different soil properties. That's why we couldn't get the same data so far. But since this research is a long-term monitoring, we are planning to collect the samples from next year, and also even though I'm not sure whether I'm here or not at the moment, but someone will collect the samples from the one cycle after one cycle or 2 cycles of the rotations, and that someone will compare the data with the data provided from the KBS, and also data from the Lux Arbor I observed. Also, we are planning to collect the one more set of the samples from the center of the crop field, which was 20 meters apart from the prairie strips, to compare the volume of the pores, and also to compare the carbon content with the sampling location close to the prairie strip. And we're also planning to measure other carbon characteristics, such as the particular organic matter and mineral associated organic matter, also as well as some other soil pore characteristics using our scan images. So this is end of my part, and we have another nice research.
Before we go on, Jinho, what's that?
I was just asking if you're saving questions.
Yeah. No, before we go on, does anyone have any questions for Jinho? You can put them in the chat, or definitely you're welcome to unmute and talk as well. Or you can raise your hand. Yeah, go ahead, Corinne.
Yeah.
This is so cool, and I'm studying microbes in the prairie strip and the surrounding cropland. I'm a grad student at Sarah Evans. I think we met before.
Yeah.
But I was wondering, like, with your work looking at pore structure in the prairie strips and the surrounding crop, do you know if there are particular prairie plant species that contribute root architecture that leads to those beneficial pore sizes? Because prairie plants grow in all different ways underground, so are there certain plant species that form those beneficial pores?
we haven't measured the different pore structures by the different plant species, but we did some greenhouse experiments using the three plant species: switchgrass, 1,4, which is a really fine roots, but extend more, like Susan, and bush clover, just like you. And if we are thinking that the form, the black eye Susan tend to create the more the greater amount of the finer roots compared to other grasses, and that size of the root should be correspond to the particular size of what we are interested in. So yeah, maybe the plant species with having the fine roots will be helpful for our carbon gains.
You know, your diagrams are amazing. The sequences up there, I love them. It's very easy to follow.
Oh, thank you.
If you said this, forgive me, but how deep are the cores that you're taking?
Oh, I missed it. Yeah. We collected the cores and the roosters from 5 to 10 centimeters. Then.
Saman had a question.
I have a question, quick question. So it's very interesting study, plus a very visuals are really great. Okay, so my question is, it's not really for the study for the future research. So, like, how about the note? So does it have any impact on the soil health or the kind of parameters to measure? If we have a chance, like we could consider P2 treatment, which has no tip.
Should be really interesting because the prairie strip and the switchgrass is technically the no-till treatment. So we can compare the no-till crop with the no-till the perennial vegetations. Then reasons, but sadly, we haven't had the chance to collect the sample from P2 to no-till plots, right? It's really interesting. Yeah.
All right, sorry, Kareem, we'll go to YJ and then we'll.
Thank you. Yeah, this is a very interesting research, and I have a couple of questions. One is that what's the mechanism you decide to, and you know that the pore at the size 50 to 150 micrometer is the critical range to help the microbiome or the process? This is one question. I think this is very important for me.
That size of the pores is known to have known to provide the nice habitats for the microbes. If the pore size is finer than that particular range of the size, then the microorganisms are hard to live and thrive in there. And also, if it's larger than that size of the pores, then because of the the continuous water flow, and because of it's too spacious for the microorganisms, then so that's why we decided to look at the specific pore range, and also indeed we found the greater the enzyme activities from that certain size of the pore range. Actually, Sasha found that, and also greater the mass of the microorganisms should be living in that particular size. So we are assuming that habitats, that nice habitats of the microorganisms should work hard in the carbon processing, like decomposition of the organic matter, and also other beneficial processes. Even though I'm just looking at the carbons, but it will be also helpful for the nitrogen cycle. It has also good in the nitrogen cycle as well.
We'll move on now, but the Sasha's lab will be here after the meeting for a little bit, so please hold questions and be sure to discuss with them afterwards.
No, I'm Gautam Thodakovi, first year graduate student from Kravchenko lab. Today I'll be talking about the project that I'm working from LTER. So that as you are all familiar with REX called the rain exclusion experiment. And to just talk briefly about the background, the project was deployed in the growing season of year 2021, and the rain exclusion of this project is achieved by setting up the rainout shelters of the three plot level treatments of MCSC. Those are T1, T2, and T7. And within those three plot level treatments, three shelters are established to have control over the precipitation. So they are controlled, drought, and variable. And I think you pretty much know about the details of each of the precipitation treatments. And under the each shelter, we do have 4 subplots. So the subplots are to look at we have sub-treatments of various carbon sources and to manipulate the diversity. And the carbon sources are addition of biochar, switchgrass, and sorghum. And whereas to manipulate the diversity, we have the fungicide and nematicide treatment. So, and as we all know that the REX primary goal is to examine the effects of resources and the diversity on resilience to the water stress. And with being that my specific research questions to this project are, so what role does fungi and nematodes play in soil pore structure and pore hydraulic connectivity at micro and macro spatial scales? So to address this question, and we have an objective to assess the effects of biological diversity manipulation, that is the presence of fungi and nematodes on soil micro and macro scale physics and hydrology, including the soil pore structure within the intact soil. So the next research question that we framed was to how the pore structure and hydraulic connectivity influences the accessibility of soil organic carbon to the hydrolytic enzymes produced by the microbes. And the third research question brings the opportunity to actually collaborate with Dr. Evans and lab. So to look at how the changes in pore structure are related to the microbial community composition, microbial function, and spatial patterns in the microbial function. So with these research questions framed and the objectives that we have, so we collected soil core samples on July 27th of 2022. And so actually from each shelter, we collected 4 soil cores from each subplot. And as you can see here, from the center of each subplot. And to locate the center of the subplot, we used the coordinate systems to locate the subplots, and as the center point was not established during the time. And so for that, we took advantage of the PVC pipes that has markings with equal intervals to locate the center of the plot. And we collected the soil cores from 5 to 10 centimeters of soil depth with 5 centimeters in height and 5 centimeters in diameter. And we also collected the loose soil for about 50 to 100 grams from the same spot. And as of now, the research plan to analyze various properties of soil. So under the biological properties, we do have the soil microbial biomass that is measured. And so the soil microbial biomass is an important factor in the nutrient cycling. And yes, and also it improves the pool of the available nutrients. And the second is the activity of hydrolytic enzymes from the microbial biomass, and which is ongoing now. So this again helps in the decomposition and aids in the mineralization. So of the chemical properties, we studied the total organic carbon and nitrogen from the samples. So the properties that are mentioned in the red will be studied in near future to study the effects of the carbon sources and diversity manipulations. So of this we have ergosterol quantification. So ergosterol is substance specific to the fungal cell walls, and so this estimation gives the abundance of the fungi. So as we have the diversity manipulation to that aids in to study the abundance of this. And of the physical properties, we will be studying the pore size distribution, porosity, and water retention. So firstly, we calculated the gravimetric moisture content from the 3 precipitation treatments, and obviously the results here reflects the effective functioning of the Raynaud shelters, as we have statistically significant low moisture content for the drought treatments. And so this is to be noted that the samples were collected on July 2027, and this is a gravimetric moisture content as of that time. So this slide here shows the soil organic carbon percentage of the total subplots, precipitation treatments, and also the main treatments of T1, T2, and T7. And it should be noted that we do have different subplots in T1, that is the conventional till, and different subplots to T2 and T7. In T1, we do have subplots of biochar, control, and sorghum and switchgrass as a carbon sources. And whereas in T2, T7, we have sorghum, fungicide, and nematicide as a subplots. So the results here shows that the subplots with added biochar has high soil organic carbon percentage, and in T1 treatments. But whereas in T2, the nematicide treatments subplots has high soil organic carbon percentage, but the reason for that is yet to be studied. So here the bar graph shows the soil carbon percentage for the 2 subplots that are common for T1, T2, and T7. And so as we look at 3 different precipitation treatments, the addition of sorghum to drought plots has actually increased the soil carbon percentage. And it is same with nitrogen percentage as well, with the addition of sorghum to the drought plots has increased the soil nitrogen percentage. And we also measured the microbial biomass, and this bar graph shows the same as the 2 subplots that are common for T1, T2, and T7. And it's interesting to note that the dissolved organic carbon with the addition of sorghum in the drought plots has increased the dissolved organic carbon, and the trend is same with the soil organic carbon, which has increased carbon percentage with sorghum. So we did also analysis for the enzyme assays for the hydrolytic enzymes, and these are the two enzymes of beta glucosidase and phosphatase. And for now we did not find any statistical differences of these two. And the analysis was performed only for the subplots of control treatment as of now, and the work is still ongoing. So I think maybe because of the variability in the data. So these 2 images are from the of X-ray micro CT scanning, not from the Rex plots, but from a different project. So these 2 images are actually from the same soil core sample, and we do have another project going on with different species of plants that are growing in the greenhouse. And the image on the left is from the sample from the core, of which we placed the soil core in the ingrowth pot, and this is before placing the soil core in the ingrowth pot, and this is after. This is the scanning image after the ingrowth pots were grown, and then we took the scanning image again to see the differences. So here we can see the difference in soil pore structure, how the growth of those plants in the greenhouses has affected the soil pore structure. The intent of these images is to show how effective is the X-ray micro CT scanning to look at the pore size structure and study the porosity and pore size distribution. So the same will be the plan is to scan the images for the Rex collected samples as well. So with this, we are hoping for interesting results to draw any conclusions. So that's all I have for now, and I would like to thank Maxwell for helping me out in sampling, and Namir for the sampling procedure, and to all of you for setting up the rainout shelters. Thank you.
Thank you, Gautam. Are there any questions for Gautam before we keep going? We are running up on time, but I want to make sure we leave some time.
Hi, this is Tavisha. I just had a quick comment. I just wanted to say that I think it'll be really cool to mesh our data together. I'm doing the PhD student in the Sprunger lab doing a lot of the nematode work, especially with the nematicide plots, and I think it's really cool that you're finding like higher soil carbon within the nematicide plots, and from like initial data analyses and like just like counts and identification, we're actually finding kind of more bacteriivores in those plots that has kind of sometimes been linked to higher soil carbon sequestration. So I think that that's really, really neat, and I can't wait to talk more about this.
Yeah, that would be really great. I look forward to talking to you.
Awesome. Great. Yeah, YJ. Go ahead.
I just have a question. The high carbon, soil carbon content in biochar, is that just the same form of the biochar we add, or it changed to different type or something?
I think it's just because of the form that we added to the subplots for now. So I just got the results recently, and I had to study more on this.
It's nice to see that it didn't all blow away when we were incorporating it. Stuck around. Okay, cool. If there are no more questions, then we will continue on.
Hello everyone. My name is Paulimi and I work in the Kravchenko lab, and most of my work deals with oxygen in soil. So as we know that I basically try to see how the oxygen distribution in soil is affected by the various pore structure. As it has already been established that the soil pore structure plays a very important role to know how the microbial activity will go on in the soil. But the first thing that the soil pore structure will affect is the oxygen availability. And through oxygen availability, a lot of other processes in soil will get affected. So basically, I'm trying to understand the microscale processes in soil that affect, and the microscale processes, how the pore size affects the oxygen consumption by the microbes. And because it's very important to go into those processes, as we can see that some soil or some treatments are releasing less nitrous oxide, for example, and sequestrating more carbon. And it might be knowing these processes, we can answer to some of the questions why this is happening. So this is the instrument that I use, and it's like a motorized microprofiling system, and we obtained it from Unisense. So this is the microsensor. It's a Clark type oxygen microsensor, and the precision is very good. The tip can go from 2 micrometer to 200 micrometers. And then we have a stand, which is a micromanipulator, and it has a step size that can go from 1 to 1000 micrometers, and it can go to the 5 centimeter to do the soil profiling. And so basically we can get oxygen at every 1 micrometer of our soil to the depth of 5 centimeter, which is pretty cool. And then we have a magnifying camera to set the whole system, and then we take the profile in our soil. So it's straightforward and simple setup that I use for my experiments. And so when I got this setup, I tried to see how the oxygen in sand would look like as compared to the one in soil. So I packed some small cylinders, and then I put some deoxygenated water in it, and I tried profiling them. So this is how it looks like in sand. since it was a deoxygenated water, so at the first profile that I collected, there was some depletion in oxygen. However, when I took subsequent measurements, there were oxygen in the soil, in the sand, and it's like Ottawa sand that I have used for this experiment. So it's like pure silica and it doesn't have any microbes in it. And we tried to not contaminate it while doing the experiment. So there was no oxygen. Even after 44 hours, there is no oxygen depletion. There was a lot of oxygen in the sand. However, when I tried the same with soil of the same fraction, we can see that there is a depletion of oxygen with time, and that's because the soil is respiring and the microbes are eating up all those oxygens and carbon dioxide is being produced. And so this gave us like, I thought like whether this thing would be affected by the different pore sizes or not. And so, and this is what we get in soil. After some time, it becomes anoxic. And so at that stage, the oxygen transport is diffusion limited and it reaches a steady state. So this gives us an opportunity to solve the diffusion equation, and where the parameter Rz is basically the volumetric oxygen consumption. So my study would focus on getting those oxygen consumption rates in soil with contrasting pore structure to kind of quantify how the pore structure is affecting the microbial activity, and it's a feedback mechanism. Also, in one of my experiment, I tried to put a leaf in the soil and see how it behaves. So for this, I didn't completely saturated the soil. I kept it at -5 kPa and I took measurements. So this green is the leaf, and I tried to get the profile at just the edge of the leaf, a bit farther, like 0.5 centimeter farther from it and 1 centimeter farther from it. And this is how the profile looks like. So the green one is the closest to the leaf and it had the maximum depletion in oxygen as compared to the yellow one and the orange one. So yeah, like this is a very preliminary experiment, but it does tell us that when soil where there is some debris or the leaf or root gets decomposed in the soil, it affects the oxygen, and we're trying to quantify how they are getting affected. So LTR has many long-term experiments, and I would like to know how the soil oxygen in the soils are. So yeah, that's it. That's what I'm doing. Thank you.
Are there any questions for Olimi? So in this one, is the leaf is essentially could be considered to be on the left, like to the left of the graph, and that the closer you are, the green is the closest, the yellow is the next closest. That's really awesome to be able to visualize that. What are the limitations in terms of when you're repeatedly sampling the soil? Are you not disturbing it by moving your probe around? Does it not deteriorate the core over time to get repeated measurements from it? Or?
Yeah, so we try to make different replicates so that we can get one measurement in one core.
Okay.
And it's a micro sensor. It's pretty thin. We try to get the thinnest one possible, and so minimal disturbance.
That's very cool. Yeah, I've never seen data like that. So that's all right. If there, let me just check what time we're messing with right here. Okay, yeah, we are at 10:30, so we can call it there. But let's thank the Kravchenko lab for sharing all this awesome.